So we are going to finish chapter 7, and then we're going to move on to, not chapter 7, are we in chapter 7? Or chapter 7, we're going to move on to chapter 8, which is Mendelian genetics. Okay, so you'll remember from our discussion in lab last week that chromosomes are chromatin that has been tightly packaged. And so it has been tightly wrapped around histone proteins. And so this is a diagram that shows the DNA, and then it shows the histone proteins. And chromosomes have characteristic size and shape and banding patterns. And so we have what is referred to as a picture of the karyotype, of the picture of the chromosomes, and this is called a karyotype. And so a karyotype analysis looks at the chromosomes that are taken from an individual. The geneticist who does this would then pair up the chromosomes based upon their size and their shape. So chromosomes are paired up based on the size, the shape, and the staining pattern, which is sometimes referred to as a banding pattern. So they stain the chromosomes, and where there is more genetic material, where it's more tightly wound, it will come out to be darker. And when there's less genetic material, where it's more loosely wound, it will become lighter in color. And so sometimes this is done to fetal cells. So for example, if you have a female who's like maybe 30, late 30s, early 40s, and she's pregnant, they might take a sample of the fetal cells. So what is it called when you take a sample of the fetal cells? Does anybody know? How do you get the fetal cells, typically? Yes, so you have to take fluid out of the uterus. And so that is referred to as an amniocentesis. Right? And so this is, they stick a needle through the abdominal wall and into the uterus. And as they do this, they have an ultrasound technician watching the baby, right? And so they stick it, and you can see the needle go right into the belly. And then what they're doing is they're drawing out amniotic fluid with fetal cells. So this is a way to obtain amniotic fluid with fetal cells. So I had this done with my second child. It was very interesting to watch because I could see the needle go in because of the ultrasound, and the baby actually like went towards the needle. It's like, what is that? You know, but obviously they don't want to actually prick the baby, so they're kind of worried about that. So then they draw just the amniotic fluid that has fetal cells. Then they take those fetal cells and they allow them to replicate in the lab, and then they get the DNA out and they stain the chromosomes, and then you get a picture. So the geneticist actually sent me a picture of the chromosomes of my child, and it looked very similar to this. And so here you can see that we have 23 total pairs of chromosomes, although here the last pair is not homologous. And so would this be a male or a female person? This is a male because they have an X and a Y. If there were two chromosomes that were the same size and the same shape and had the same banding pattern, that would be a female. Okay, so this is what a karyotype looks like. So this is chromosome number 1, and notice how much bigger it is than chromosome number 22. So they pair them up based upon their size, and they don't, you know, the geneticist actually has to cut and kind of paste the pictures together, right? They don't come out all grouped like this, right? So the geneticist has like a search image, and she can quickly pair chromosomes up because she knows exactly what they've looked at because she's looked at so many of them, right? Or he has looked at so many of them, okay? So we have what are said to be 1 through 22 pairs of autosomes. So that is an autosome is something that is not a sex chromosome. So we have 1 pair of sex chromosomes. And if you have 2 X's, this is said to be the homogametic sex. So they have homo means same, and so they have the same chromosome in their, so they have 2 copies of the X chromosome. If you are XY, that is the heterogametic sex. So hetero, that's an E, heterogametic sex. Okay, so this one would be female, and this one would be the male symbol there. Okay, so if we look at another example of a karyotype, this is kind of a modern version, which is, they didn't do it for me, what, 10 years ago, but it might be what you would see, where they actually, instead of, it makes it easier to actually paint the chromosomes. So they add a fluorescent probe that has a particular color, and then it just attaches to the genetic material on that particular chromosome, and so that they come out looking like this. So you can see it would be much easier to match these up than the black and white picture. And so this individual would be what? Male or female? Female, right? Because it is the next chromosome. Okay, well, sometimes there are mistakes being made when the chromosomes separate during meiosis. And so we're going to talk about those errors in meiosis. So if you have the correct number of chromosomes, you are said to be a euploid. Okay, so U means good, ploid means copy. So that means that you have the correct number of chromosomes, and that is 46, right? So that would be the correct number. So if you do not have the correct number of chromosomes, you put an A in front of that, but AEU, how do you pronounce that? So they put an N. So it's called an aneuploid. And so this is not the correct number of chromosomes. Because A means without. So without a good copy. Okay. Now in animals and in humans in particular, we do not see a lot of examples of aneuploids because in most cases it would be lethal. So for example, if you had an extra copy of the first chromosome, so you had like trisomy 1, then what that would mean would be that you would have a lot of extra genetic material and the baby or the embryo probably wouldn't ever live long enough to implant, right? So if you have 3 copies of a chromosome, it is said to be trisomy. Right? So that means that you have 3 bodies, and the common ones that we see are 21, we see trisomy 18, and we see trisomy, sometimes trisomy 13. Yes?
This would be the sex cell of the parent.
This would be in the karyotype of the baby, right? But it's happening, it has, so if I'm trisomy 21, that would mean I have Down syndrome. But it was due to an error in meiosis. So we'll talk about what's happening in the sex cell, okay? So the point here is that it doesn't occur any more commonly to get trisomy 2, 4, 6. We just don't see them, right? So these are the ones that can survive, that where it is not lethal, okay? We can also talk about monosomy. So that would be only one cell, right? And monosomy, and so these are autosomes right now. I'll talk about sex chromosomes in a minute. But monosomy is where you are missing, so you only have one chromosome. And so this is only in the sex chromosome. So this is an individual that has one X chromosome, and that 0 means that they don't have another one. So they don't have another X, and they don't have another Y, they just have one X. And so this is what is referred to as a sex aneuploid. And it has a name, it's called Turner's syndrome. So people can survive with this and they can be fairly normal, but they generally have infertility because of this. So they are female from external characteristics, but they tend to not be fertile. And there are also other sex aneuploids that I forgot to mention. So I'll list them down here when I was talking about trisomies. So we can have XXX. That is okay. Oftentimes we might not even know that the person has that. I can't remember what they call that. Okay. We could have XYY. This causes some, actually this doesn't seem to cause any problems. I think it's called Jacobs. This is Jacobs. So Jacobs syndrome. Yes?
What is prominent in hermaphrodites?
Hermaphrodites could be chimeras, which means that they actually have 2 different types of cell lineages in them. So maybe we'll talk about that later when we get to development, embryonic development. Okay, so Jacobs disease is, this one is a really interesting one. Has an interesting history because when they discovered this, they thought, oh, if a male has an extra Y chromosome, that might mean that he is extra aggressive, right? They thought, oh, it does tend to cause men to be bigger, and they tend to have really big hands and feet. So that's one of the phenotypic effects of this. So the idea is that perhaps people in prison were more likely, or males in prison were more likely to have this genotype, so this combination of chromosomes, than people outside. And they did this study, and in the 60s, I think it was in the 60s, they published it. And it caused this huge uproar because they promoted actually having baby boys tested. So when you had a baby boy, they would be tested, every boy would be tested, and if they had a Y chromosome, then they might be treated differently, right? And that is like kind of counter to how we believe in, you know, that the environment, not the genes, determine who the person is for some parts, right? So it actually showed that initial publication was flawed, and that is because we didn't have a good sample of the general population outside of the prison. So it isn't any more likely that male inmates would have this genotype than you would find them in the regular population. So once they did a valid study, which took into consideration a bigger outside population, there wasn't any difference. And so we do not, you know, test for that. And it would be like, I mean, I wouldn't, if I had a son, I wouldn't want them tested because, you know, they might be treated differently for the rest of their lives if people knew that they had an extra Y chromosome. Okay, so that one is interesting. There is one that is XXY, and this is called Klinefelter's. And that one does cause infertility. This individual would look male. Sexually, the external genitalia would look male. Right? But they would also have infertility problems. So like Turner syndrome, they might have some other problems associated with having that X chromosome. So really with Turner syndrome and Klinefelter's, we generally find those individuals in the population because they go to the doctor and the doctor says, oh, there's something different and maybe possibly happening with this person, and then they would be genetically tested as children or as young adults. Yeah, Morgan?
How do you know or test for infertility? Like, there's no like.
Well, they probably would, like females wouldn't get their periods, so they would never get their period. And so then they would do a test and they would find out that their ovaries are not producing, you know, estrogen, or they're not developing eggs.
So
They're not ovulating. Yeah, there's symptoms.
Oh.
Yeah. Okay, but that's a good question, actually. Okay, so those are examples of the aneuploids, the incorrect number. And so if we looked at how this happens during meiosis, we have a process that is called, so we'll look at the mechanism. And this mechanism is called nondisjunction. So nondisjunction is where the chromosomes do not separate properly by or during meiosis. So you'll see here that this is a gamete, and it has a pair of chromosomes where it shouldn't, right? So this blue one and this red one are actually homologous chromosomes. This one would just have one chromosome, right? So it would be missing the chromosome. And so if these came together, so like for example, if these two came together, there would be the incorrect number of chromosomes. And nondisjunction can occur in meiosis 1, but it can also occur in meiosis 2. So the question is, when we talk about Down syndrome, which is trisomy 21, that means they have 3 copies of the 21st chromosome. Why does that tend to be more common in females as they get older? So as you get older, your risk of having a baby with Down syndrome increases dramatically. So there's a couple of hypotheses for this. One is that the eggs are stored in the ovaries in prophase 1. So when you are born, your eggs are in prophase 1, and they stay there until they are signaled to develop. And so they might stay there for 37 years in prophase 1, and maybe the age of the egg has something to do with whether or not it is likely to produce a haploid cell, a gamete, that has the incorrect number. Males are not that way. So male cells, they start out as a diploid, and then they divide, and one of the daughter cells will undergo meiosis, and so they're continually producing new gametes. They don't hold their gametes for a long period of time in meiosis. So that's the one hypothesis. The other hypothesis is kind of interesting in that trisomy 21. This is a pretty viable embryo, and perhaps the mother does not screen out the embryos as thoroughly. I'm thinking not, she doesn't look at them, but her body doesn't screen them as thoroughly when she gets older, because her chance of getting pregnant diminishes as she ages. And so these embryos that have this can kind of sneak past that screen, because all embryos actually have to pass a test in order to be maintained in the uterus. And that test is that they have to produce a hormone, and the hormone is called HCG. And if they don't produce enough HCG, then they are going to be spontaneously aborted, right? So there's a couple of hypotheses as to why it is more common in older females to have this problem, okay? So if we looked at a karyotype of a person that was a person with Down syndrome, it would look like this. And there is a specific notation, set of notation for this. So I'm going to write my notation because I'm going to give you homework where you have to figure out what the notation is. The notation is that I have 47 chromosomes. So if I counted up all those chromosomes, because I have an extra 21st, there should be 47 total. And then I'm going to put the sex. So this one obviously is a female. And then I'm going to put plus 21. So that is an individual that is a female with an extra 21st chromosome. So that's how I would write the notation for that, for this particular karyotype.
Okay, so it's not with an additional 21 chromosomes.
No, so it's that plus 21 means it's the 21st chromosome that's extra. Okay. If there was the 13th chromosome, it would be plus 13. If it was 18, it'd be plus 18. If it was 1, it'd be plus 1, right?
So would they still have Down syndrome if it was in plus 13?
No, it would be a different syndrome. Yep. So different trisomies give different phenotypic results.
And not all of them are physically noticeable.
All of these are. If you had, yeah, so all of those are.
So what syndrome would be 13 and 18?
You're going to find out. At least the 13th one you will. Okay, so, and your homework. I'm going to give you homework. Okay, so there is something else that happens, and this is called deletion of a piece of the chromosome. So the chromosomes can break during meiosis. As they are being separated, they can break. And so this is a karyotype of a person that is missing parts of one chromosome number 5. So this would be a deletion on chromosome number 5. And this is probably the most common deletion that we see. Again, if a chromosome breaks dramatically, the embryo will not be viable. But this is a little bit of a deleted region. And so they actually show it in more right here. Okay. So this produces a syndrome called Cri-du-Chat. And the reason for this is that one of the phenotypic effects of having this deletion is that their larynx does not develop properly. And so they sound like a cat when they cry. And so it is one of the most common syndromes. So 1 in 20,000 to 1 in 50,000 babies might have that, probably depending upon the population.
Like a literal cat.
They cry like a cat. It sounds like a cat when they cry. They have other phenotypic effects. And so they're usually small at birth, right? They have respiratory problems. The larynx doesn't develop correctly. They have distinctive features like a small head, unusually round face, a small chin, widely set eyes, okay? And then they also have problems with their heart and their muscle, and then they don't develop their nervous system properly. So that is a chromosomal deletion that causes changes in the phenotype. Okay. So we're going to change topics a little, and we're going to talk about the cell cycle. So I mentioned before, and you had a picture of your cell cycle in lab. So we talked a little bit about the cell cycle, but there are different stages. So there is, for example, G1, and G stands for, you know, I thought it stood for growth, but it can also stand for gap, I guess. So this is growth, and G1 occurs right after a cell divides. Okay, so this occurs right after a cell divides into 2 daughter cells. So if we look at our diagram, what does M stand for right here? What do you think it stands for? Mitosis, right? So that is asexual reproduction. That is what is occurring in our cells in our body, the somatic cells, for example. But you'll notice here that G1, after G1, cells could go out of the cell cycle. They could leave the cell cycle and they could go into G sub 0, okay? This is the stage that is outside the cell cycle, and this occurs when cells specialize, right? So this is leaves the cycle and becomes specialized. So for example, neurons typically develop and they're produced, and then they leave the cell cycle and they become specialized, as do skeletal muscle cells, smooth muscle cells, cardiac muscle cells. And so the question that we have as scientists in the medical field is, how do we get specialized cells to start reproducing again in order to repair damage? So how do you get cells that, like in the spinal cord, if the spinal cord is severed, how do you get cells to replenish themselves, right, to regrow and go back into the cell cycle so that they can repair the damage? So G2 is the that. And then we have S, and S stands for what? Do you remember from lab? Synthesis, right? So this is synthesis. So this is where DNA replication occurs. So once it has replicated its genetic material, it pretty much is on the road to dividing again, right? So G2 occurs right before cell division. So everything that is needed, all the proteins and the spindle fibers, everything that is needed for cell reproduction occurs at G2. Okay, so what we need to know is what regulates the cell cycle, and we need to know that because cells like cancer cells do not follow it, right? They do not obey the signals to stop dividing. So we can talk about, we can talk about the regulation of the cell cycle. Okay. So we have external factors. And one big external factors would include hormones. So for example, estrogen. So estrogen is known to promote cell division. So it causes cells to divide when it is present, and specifically cells that line the uterus, and also cells that are in the breast tissue of females. So when a female is getting ready to ovulate, the lining of the uterus thickens due to mitosis, and also the breasts swell due to mitosis. And then if pregnancy does not happen, then the cells die and the tissues will shrink back down. So estrogen promotes cell division. Now, interestingly, we have lots of synthetic chemicals in our environment that seem to mimic the effects of estrogen. So kind of as an aside here, right, we have synthetic pollutants that mimic estrogen. And we see that there are, in some cases of animals, we see that estrogen, these estrogen mimicking chemicals get in the water and it messes with their reproductive development. So specifically like amphibians, like frogs, right? They might be exposed to atrazine, and atrazine affects their ability to develop into a male because they're being exposed to all these estrogens in the water. And the same thing with the alligators. Alligators are being produced that don't have, the males have tiny, tiny little genitalia that doesn't function properly. And so you see a decline in those populations as well. But there is one that you probably have heard of because it was a huge deal. This was a big deal in the scientific community and politically. And this is BPA. Where is BPA? Plastic, right? So this leaches from plastic. So the female scientist that discovered this, she was actually looking at cells in test tubes. And, you know, scientists use plastic a lot. We use plastic test tubes. And she was noticing that it didn't matter if she added estrogen to them, that the cells in the plastic containers were undergoing cell division regularly. And so she hypothesized that had to do with something coming out of the plastic test tube and causing the cells to divide. And when she published this information, it was huge. And she got this huge backlash from the plastic industry. And she was just essentially almost like her career was almost destroyed overnight over this. And so it is a big deal that we now have lots of plastic that is labeled BPA free. And I can't remember her name, sorry. But it's all thanks to her, right? So we now have water bottles BPA free, and we now don't let our babies chew on plastic toys that leach BPA, right? And it's not in babies' bottles, right? And that is probably a really good thing, because that could lead to infertility in the babies that are exposed to BPA. Their reproductive organs would not develop properly. Okay, so that is estrogen. We have another one that is called growth hormone. And interestingly, it is not a steroid, so it's not a lipid, it's actually a protein. And growth hormone affects the bones. So it causes growth of the skeleton. Right? So the whole way that we can lengthen our skeleton as we grow in childhood is due to the presence of growth hormone causing the cartilage in the epiphyseal plates, in the growth plates, to undergo mitosis. Right? So those are some external factors. Okay, internal factors. So internally, we have some proteins called cyclin. So I like that name because it's easy to remember, right? Cyclin proteins. And there's different kinds. There's actually 4 different kinds, and they just labeled them A, B, there's no C, D, and E. And what these cyclin proteins do is that they accumulate in different parts of the cell cycle, and their presence in that part of the cell cycle allows that cycle to continue, okay? So if the appropriate cyclin protein is present, the cell cycle continues. Okay, if the protein is absent, right, the cell cycle stops. So we learned about tumor suppressor proteins. They, like P53, is an important molecule gene because it will actually stop the cell cycle. So it would prevent the cyclin from accumulating, and it will stop the cell cycle. So if the DNA is damaged, it will prevent the cell from undergoing cell division, okay? So in your book, you don't need to know the details about this, but notice how there are little checkpoints along the way, and these checkpoints correspond to a particular cyclin being present, okay? So for example, after the genetic material is replicated, cyclin A has to be present in a relative amount of abundance in order for the cell cycle to continue. So that is one internal mechanism for how we regulate if a cell is going to stop or if it's going to continue to be allowed to be able to reproduce. Kind of, I think, a more interesting mechanism, which is very interesting from the perspective of aging as well, is what is called the mitotic clock. Now, this doesn't determine when a cell is going to stop dividing, but it determines how many times a cell will be allowed to develop or divide. So it determines how many times a cell can undergo mitosis. So the mitotic clock molecularly is the ends of the chromosomes. So remember that the center of the chromosome is called a centromere. At the ends of the chromosomes, we have what are called telomeres. So the telomeres are highly repetitive DNA that don't really code for anything. So it's not significant if we lose them. And so what happens is every time the cell undergoes mitosis, it loses some of the DNA off the ends of its chromosomes. So it's counting down, and when it has no more telomeres present, it will stop dividing, right? So every time a cell divides, it loses some of its telomere. That's every time a cell divides. Divides. Okay, so when there's no more telomere present, it's telomere. When there's no more telomere present, then it can't divide anymore. Now, as I mentioned in the video, there is an enzyme called telomerase. And does anybody remember what I said telomerase does? It adds, right, DNA back to the telomere. So it's like going back in time, right? So you can have an enzyme, this enzyme is turned on and adds the telomeres back up, then the chromosomes are of a young age again. Okay? It's time reversal. So one of the interesting things is that cancer cells turn on this gene. So that means that tumor cells, for example, have been around for, 67 years about, and they're still dividing, right? They're still undergoing rapid cell division because they have no mitotic clock. The cells that give rise to your egg and sperm have this turned on, right? Because you don't want the egg and the sperm having shortened telomeres, because that will shorten the lifespan of your offspring, right? So it would be turned on in cancer cells and cells that give rise to egg and sperm.
Something synthesized or something like
This is something that our body creates. We have this gene. It's normally turned off in most cells, but it's turned on in cancer cells and cells that give rise to the gametes. Now, what they discovered is that it might actually be turned on elsewise in other times. So the size of the telomeres seems to be related to your environment. Okay, so stress, right, speeds up the mitotic clock. So how stressed you are will cause your telomeres to shorten faster. So you might, notice this about people that go through really bad, stressful situations. It's like if they go through a really bad, stressful situation and like a year later, it looks like they're 10 years older. And you're like, whoa, that person aged really fast, right? It probably has to do with their telomeres. Because if the telomeres shorten, then their cells can't divide and they can't replace damaged tissue. And so they start to look old, right? Their hair falls out, for example, right? So stress, right? Nutrition, right? Drug use, okay. All those things can speed it up, but they've discovered that some things can reverse it, right? So if you eat a good food, if you switch your diet, you get off the drugs and you reduce your stress, your telomeres will actually start to lengthen again. So it's kind of interesting because there is now a biotech company trying to make money off of this. So you can have your telomeres tested. I don't want to sign up for this. Okay. So this is just one company. There are other companies. And so this is the telo years. And so what they want to do is they want to get a sample of your genetic material and before you do something dramatic, like change your lifestyle, go on a diet, get some exercise, and then they want to see how it changes over time. And so that you can pay them to do this for you.
We're giving them
Your cells at the ends of your DNA are protected caps called telomeres. They shorten with age, but also change with certain lifestyles, both good and bad. Go beyond ordinary vital signs to monitor the vitality of your cells with telomeres, a simple genetic test that lets you learn your cellular age based on your telomere length. There are steps you can take to slow down the clock on the aging process, but if you don't measure it, you can't manage it. Control how well you're aging. Learn more at telomeres.com.
Yeah, cute little telomeres, chromosomes. Okay, so you can be tested to see if your telomeres have artificially shortened. And they've actually done interesting studies with children that were in really stressful childhood environments. And they do note that their telomeres are shortened compared to children that didn't experience a lot of childhood trauma. I don't know how much this one costs.
The fact that we have to pay them is kind of dumb. Like they're collecting this.
Sorry, I don't know how much it costs.
They are gonna put the price on the website.
Yeah, I'm sorry, I don't know. Okay. Okay. Okay, so that would be regulating cell cycle, when the cells can continue to divide and when they have to stop. So the next topic that we're going to go over, and we're going to actually work on this in lab, is Mendelian genetics. And so this is a new section of the class, which involves a little bit of problem solving. And so some people love it, some people they do not like it at all. Okay?
This is chapter 8.
This is chapter 8. So most of you might know this is a person named Gregor Mendel. And he was around in the 1850s. And he was interested in testing another hypothesis that was around about how traits are passed from parent to offspring. And so he tested the blending theory for the pattern of inheritance. No. So the blending theory was the theory at the time for how traits were passed from parents to offspring, and it kind of makes sense when you look at it from a big picture. Because the blending theory stated two things. One, that offspring are always a blend of the parents. And specifically, they're kind of intermediate in the color. So it could be like, they would always be an intermediate color between, if you're talking about skin color, between parents or offspring, or hair color or eye color. They would always be a blend of the parents, right? So that kind of makes sense. The second thing is that once blended, traits cannot be separated back out. Yes. So once blended, we'll put the characteristics. Can't be separated back out. And that kind of makes sense, except for the fact that you can imagine if this was true, that we would all kind of move towards some kind of intermediate, and so we all start to kind of look alike. And so this doesn't explain this particularly, doesn't explain how variation is maintained from one generation to the next, okay? So this was the theory prior to Mendel. So this is the theory that he tested, right? And so let's look at what he did to test this particular theory. Okay. So he did not use humans, he used pea plants. And he used pea plants, and he specifically chose traits that were discrete. So if you look at this, the seed shape is either round or wrinkled. Seed color is either yellow or green. So there's no greenish yellow, right? And so he was really smart to pick these traits because he probably knew in his head that the simpler the system he worked with, the better he could test out that blending theory. So if it was a real complex trait that also depended on the environment, then it's not going to be easy to test, okay? So you have to realize they had no idea what the genetic material was, what chromosomes were, what DNA was. They were just looking at putting pea plants together and seeing what arose, okay? So in the first, let's use plant height as our example. Okay. So in the first cross, he would have tall plants and he would cross them with short plants. We would say that these, in this generation, this is the parental generation. And these are true breeding seeds. So true breeding means that if I took this tall plant and I mated it with another tall plant by taking the pollen from one flower and fertilizing it, then I would always get tall plants. Right? I would never get short plants coming back out. So they're called true breeding. And you can, find a lot of seeds that we actually grow in our gardens are hybrids. They're not true breeding. Okay, so what he found when he did this is he found that all of the offspring were tall. Now, does that support the blending theory? No. Okay. So this is what is referred to as the F1 generation. Filial is the F, but you just need to know this is the F1 generation. And this would be a hybrid. It's a hybrid because it has parents that are different, right? And it is also a hybrid because it is not going to be true breeding. It is not going to always produce tall plants, okay? So if I bred two of these hybrids together, what I'm going to get is tall and short. So that does not support the blending theory either because this came back out. So once it was blended, it shouldn't come back out. So, it came back out, right? So that's why we now know that the blending theory is not the theory we use Mendelian's theory. It's actually almost probably a law now to explain how traits are passed from one generation to the next, okay? Now, these are what are referred to as tall and short. These are phenotypes, okay? What we now need to ask is what are the genotypes or combination of alleles that we would find in these different phenotypes. So the phenotype is the outward expression of the genotype. Okay, so we're going to talk about what genotype is now. So that's what they look like. It's not always external though. It could be like my phenotype could be that my blood doesn't clot, right? So I'd be hemophilia. It's not something you can see on the outside of me, but it's inside, right? My blood does not clot. That's the outer expression of my genes. Okay, so let's look at genotypes. So we have genes and we have alleles. So alleles are alternate forms of a gene. So my gene could be slightly different from your gene, and they would be two different alleles. So if I have a gene, so let's say that I have gene big A. Okay. I can have two alleles. I could have the dominant allele, or I could have the little allele. So the little a, I usually put a line over the little ones. And this is what is called the recessive allele. So I'm just assuming that there are only two versions. There could be multiple versions of gene A, but we're just interested in these two because we're looking at a very simple system, okay? So when we look at the genotypes, we can say that you could have two big A's and you would be homozygous dominant. So what that means, homozygous means you're going to produce the same zygote, right? So you would be big A, big A. I could also have two copies of the recessive allele, and I would be homozygous recessive. And so I write my genotype like that, okay? Notice that I'm diploid, right? Because our genotypes, we have two copies of every chromosome, two copies of every gene. And then over here, I'm just going to write, this is, maybe I'll write it down below. The combination is said to be heterozygous. So this would be big A, little a. Okay. So the dominant allele, in this particular instance, is going to mask the presence of the recessive allele, okay? So let's go back to our pea plants and write down the genotypes of our pea plants. Okay. True breeding, what do you think the genotype is? Big T, big T. Short, no S's, same gene, little t, little, oops, little t, F, little t, okay. Okay. Hybrids, what do you think their genotype is going to be? Okay. And then we know short is going to be little t, okay. But tall could be big T, or big T, little t. Okay. So when I'm looking at a tall plant, I wouldn't know what its genotype is unless I've read it. So there are some traits, like in pea plants, like in humans, that show this pattern. And so that's what we're going to be looking at in lab tomorrow and on Thursday. Okay, so before you leave, I'm going to hand out your homework. So this is due next Tuesday, and there'll be another quiz next Tuesday. What? There will be an online lecture.
And they're posted in a week, but probably like we're going to take that associated with this week. Okay, that's what's like.
What was that? And I slide 4.
Oh, like hang out your house, lingering in the air.
It might be.
Yeah, there are genes that expand. So expanding genes, and what they do is they make copies of themselves and make it bigger. And so it gets worse as you go from one generation to the next because the gene just gets worse.
The father of the family has white.
Yeah.
She doesn't have it.
Right. Yeah, interesting.
If you ever meet her, she's a great person to talk to.
Okay.
Have a nice day.
You too. Yep.
For the telomerase, is there like a genetic defect that could happen to it, like where it always stays short? Like in the
You wouldn't,
general-
so if it was always, like, you wouldn't be able to pass that down to the next generation
okay.
because the embryo wouldn't develop because the cells couldn't divide.
Oh, okay, okay.
Yeah, so if you had a defect in your telomerase gene, you would not, the embryo would just be, it would never even.
Okay.
Yeah.
Okay. Thank you.
Yep. It is why cloning, we'll talk about this, but cloned organisms have shortened lifespans.
Oh, okay.
How are these?
Let me get a pen. It's tricky. Okay, so like this one lines up with this,
Right.
right? But notice how this one doesn't look, it looks like it's between 27 and 25, so it actually lines up with that one. So this one lines up with this one lines up with that one, this lines up with this one. See that?
Okay.
So you could go back through and correct your test and to study.
All right. So this one would go here, that one there.
Yep. Mhm. Yep. Yeah, it's kind of weird. Must just be the way machine does it.
Okay.